Hey guys, welcome to our second lesson in our Intro to Genetics series. In the previous lesson, we looked at Mendel's work, specifically with pea plants and how he laid the foundation for much of modern genetics. And in this episode, this video, we're going to look at specifically at how we can apply his principles that he discovered to Punnett squares in order to predict what characteristics will show up when we perform certain crosses. So last time we left off here, we were looking at these things that we called alleles. And there's a couple more words that we need to add to our toolkit here. Each letter is an allele here. All right, but what do we call the pairing here? What do we call the grouping of two alleles? Well, I want to give you those terms now. And so this grouping, each combination here is referred to as a genotype. And a genotype is simply just that. It's an allele combination for one gene. It basically is just telling you what versions of the DNA for this gene does this organism possess. So that's pretty easy to remember because geno sounds a whole lot like gene. That was done on purpose. Okay, so geno refers to the gene. Well, we have another word that corresponds to what the actual characteristic is, and that word is phenotype. A phenotype is the actual physical trait that is observed. All right, so again, that's pretty easy. Pheno sounds like physical. Okay, so phenotype is the actual trait. So if the genotype, let's go up here. If the genotype is big P, the phenotype will be purple. If the genotype is little p, the phenotype will be white. So again, genotype is the gene, phenotype is the physical trait. All right, so I promised that this lesson would be about Punnett squares, so let's move on to there. A Punnett square is this right here. All right, and all it does is it's a graphic organizer just to help us make predictions about what phenotypes or characteristics will be possible in the offspring of a given cross. So in this particular cross, what we would have is we would take this genotype, big P, and write it in there, and this genotype for the other parent, little p, and write it in over there. Now, the way that it works from here is that we will just simply take this letter right here and bring it down. So big P comes and it goes down in both squares in that column. They each get a big P. And what we are showing here is this particular allele could be passed on to the offspring through sexual reproduction. And so we're showing possible combinations of offspring in these squares. This little p then will come across. Now, by convention, meaning everybody just agreed, we write the capital letter first because the capital letter is what we're used to seeing first in the English language. And so we just carried that over to this. So same thing over here, the big P, the little p. And what we'll find is from this cross, and this is exactly what Mendel observed, is that 100%, 4 out of 4 of these boxes in our Punnett square end up being what we call heterozygous. Now that's a new word. I haven't introduced you to that yet. Heterozygous means the two letters in the genotype are different. In this case, a capital and a lowercase. These again over here, both of these are homozygous. This is heterozygous. And so what happens is, because they're all heterozygous and purple is dominant, 100% of the offspring in this cross would have purple flowers. Okay? Now, you may be looking at this right now. I know some of my Scott High students are, and you're thinking, oh, wait a minute, I have kind of seen this before. You probably have, maybe in your math class. So one way that this kind of relates to math, haha, I snuck it in there. You thought you could get away with math by doing some biology work, but I'm going to sneak it back in. some math teachers would refer to this as the window method or a lattice method. And what you could do is, this is for multiplying polynomials. Now, if you don't want the extra math lesson for today, you can skip ahead a few minutes, just a couple minutes in this video, and jump back in with the biology practice. But I'm going to show this real fast. So what I would do is just take each term and write it in just the same way that we did with our genotypes in our Punnett square. And they're going to come into these boxes the same way, except because we're multiplying, that's the same operation we'll do. So x times x is x squared. X times positive 5 is positive 5x. X times positive 2 is positive 2x. And 2 times 5 is positive 10. Now what we would do is simply just combine like terms. So I'll write my x plus 2. I'll take my 5x and my plus 2x, and they become 7x together. And then last, plus 10. Now this is great because it's an alternative to the FOIL method of multiplying polynomials you were likely taught as well in algebra. But the difference is FOIL breaks down a bit when you get to larger polynomials. And so what happens if you have x plus 5 times a trinomial? What happens now? Well, you can just make a bigger window. All right, and do the same thing, x plus 5, and now each term from up here can be brought down, 7x plus 10. I won't work the full thing out, but you get the idea here. So that's a great thing, and there's your little bit of a math lesson today. You are welcome, math teachers, for helping you out. All right, but back to biology. So if you skipped ahead, here's where you want to jump back in with the biology lesson. Let's look at another example of a Punnett square in the pea plant height. So if we had big T, that would be a tall pea plant. And let's take another one and cross them together. Two heterozygous tall pea plants. And we will write in one genotype here. The other genotype we will write over here on the side. Fill in the square. Again, what's on top you bring down, what's on the side you bring over. Now, we're going to notice something interesting here. We now have all three possible combinations in this set of results. And the two big T's would obviously make that a tall plant. But right here, the big T, little t combination, heterozygous, would also be tall. So 3 out of the 4, or 75%, would be tall. And excuse me, I shouldn't say a 75% will be tall. It's a 75% probability that any offspring from this cross would be tall, and a 25% probability that any offspring from this cross would be short. And Mendel figured these things out without knowing any of the modern genetics that we do. So it's very impressive what he was able to do. So let's do one or two more examples just to kind of help you with your workbook. What about if we had, R's? Let's say the big R is for round seeds and little r is for wrinkled seeds, right? So what if we had, let's say, a heterozygous pea plant, so it would have round seeds, and let's cross it with one that has wrinkled seeds. All right, so we end up with big R, little r. Again, that is round, and in this case, 2 out of the 4, or 50%. And we end up with some that are wrinkled, the other 50%. Okay, and I kind of went off the screen a little bit there. That's all right. So what we'll do here is I am going to set up one more example, and I'm going to let you guys have an opportunity to pause the video, try one out for yourself, and then see if you get it correct, and then unpause the video And so I'll give you a chance to do that right now. Let's set up one more. Let's clear this off. We'll do one more example. What if we had, no, I don't know. What if we had, let's do color of the seeds. And we had yellow seeds and lowercase y's for green seeds. Well, what if we had heterozygous yellow with, let's do another heterozygous yellow. So they both have yellow seeds, but they're heterozygous. So you pause the video, try this cross out real fast on your own right now, and then we'll get back and you can check it with the results. Okay, so hopefully you've had a chance to pause the video and try this out. And so what we're going to do now is we're going to show the work for this. Let me move these out of the way. Oh my. Here we go. Let's move these up a little bit so we have some space to work. All right, so we will take each genotype, write it into our square, fill in the possibilities, and what we find is that we have 3 out of the 4 that have a capital Y, meaning 75% probability of it being yellow and a 25% probability of it being green. All right, well, I hope from this video that you have gotten some help understanding Punnett squares and been able to complete lesson 2 in chapter 12 of your workbook. And I would like to encourage you to check out the other videos that I have on my channel. I'll try to get videos up to help you with all of your work, maybe even go back and record some to help you with your missing work. And so if you like these videos, feel free to like them, share them with your friends at school who need help with their biology, and maybe even help me out and subscribe to the channel. So thank you guys for tuning in. Check out lesson 3 coming up next.